PCR is a laboratory technique that allows us to make millions of copies of a specific DNA sequence in a short period of time. It involves a series of temperature changes and the use of specific enzymes to carry out the reaction. It consists of three main steps: denaturation, annealing, and extension. During denaturation, a process of heating the DNA sample to a high temperature is involved, typically around 95 degrees Celsius. The high temperature breaks the hydrogen bonds between the two strands of the DNA double helix, separating them into two single strands. Then, during the annealing process, the temperature is lowered to around 50 to 65 degrees Celsius, allowing specific primers to bind the complementary sequences on each single DNA strand. Primers are short DNA sequences that are designed to match the regions flanking the target DNA sequence we want to amplify. After the primers are bound, the extension step happens. The temperature is raised around 72 degrees Celsius, which is the optimal temperature for the DNA polymerase enzyme to work. DNA polymerase adds complementary nucleotides to the single DNA strands, synthesizing new DNA strands that are complementary to the original template strands. This process extends the DNA sequence, creating two new double-stranded DNA molecules. Then, the whole cycle is repeated multiple times. Each cycle doubles the amount of DNA, resulting in an exponential increase in the number of DNA molecules with the target sequence. This allows us to quickly generate a large amount of the desired DNA sequence. Gel electrophoresis is a technique used to separate and analyze molecules such as DNA, RNA, or proteins based on their size and charge. It involves the use of an electric field to move charged molecules through a gel matrix, which acts as a sieve. The gel, made of agarose or polyacrylamide, is placed in an electrophoresis chamber with electrodes at each end, a positive anode and a negative cathode. The sample, treated with a dye for visibility, is loaded onto the gel, and an electrically conductive buffer solution covers the gel. When the electric field is applied, negatively charged molecules move towards the anode, while positively charged molecules move towards the cathode. The gel matrix slows down their movement, causing separation based on size. The separated bands are visualized by staining the gel and can be analyzed to determine the approximate size of the molecules. Gel electrophoresis has various applications in genetic testing, DNA fingerprinting, and protein analysis, contributing significantly to our understanding of molecules in biology and biochemistry. Then, after conducting the gel electrophoresis process, DNA profiling, also known as DNA fingerprinting, is conducted. It is a technique used to identify individuals based on their unique DNA patterns. After the gel electrophoresis process is used to separate DNA fragments, then amplified using the polymerase chain reaction, multiple copies of specific DNA regions are observed. These regions, known as short tandem repeats, are highly variable among individuals. Due to the varying sizes of the DNA fragments, they migrate at different rates and separate into distinct bands on the gel. These bands represent the different alleles of the STRs. After electrophoresis, the gel is stained and visualized to observe the banding pattern. By comparing the banding patterns of different individuals, it is possible to determine their genetic relatedness or identify an individual based on their unique DNA profile. The process of genetic modification typically starts with the identification and isolation of the desired gene or DNA sequence from a donor organism. This gene is then inserted into the genome of the recipient organism using techniques such as plasmid-based transformation. The inserted gene is integrated into the recipient organism's DNA, allowing it to be expressed and produce the desired trait. To ensure successful integration, selectable marker genes are often co-inserted, which enable the identification and selection of organisms that have taken up the desired gene. Now that we understand the techniques involved, let's explore some applications of genetic modification. One major area is agriculture, where genetically modified crops are designed to have desirable traits such as higher yields or resistance to pests. An example of a genetically modified crop is Bt cotton. The gene for a toxin produced by the bacterium Bacillus thuringiensis is inserted into the cotton plant's genome. This toxin is harmful to certain pests, so the modified cotton plants are able to resist insect damage without the need for excessive pesticide use. Bt cotton is an example of how genetic modification can enhance crop resistance and reduce environmental impact. Another example of a GMO is golden rice. Golden rice was developed to address vitamin A deficiency, which is a significant public health issue in many developing countries. Vitamin A deficiency can lead to severe health problems, particularly in children, as it plays a crucial role in maintaining healthy vision, supporting the immune system, and promoting normal growth and development. By incorporating beta-carotene into the rice, golden rice has the potential to provide a dietary source of vitamin A to populations who primarily rely on rice as a staple food. Cloning is a process that involves the creation of genetically identical copies of an organism. There are different methods of cloning, but let's focus on somatic cell nuclear transfer, SCNT, a commonly used technique for animal cloning. SCNT starts by selecting a somatic cell, such as a skin cell, from the organism that will be cloned. The nucleus of the somatic cell, containing the organism's genetic information, is then extracted. Next, an egg cell is obtained from a different individual of the same species. The nucleus of the egg cell is removed, creating an enucleated egg. The somatic cell nucleus is then inserted into the enucleated egg, and then the two are fused together using an electric pulse. The reconstructed egg now contains the genetic material from the somatic cell. To stimulate cell division, the egg is treated with certain chemicals or exposed to a specific environment. Once the cloned embryo reaches a suitable stage of development, it is transferred to a surrogate mother or placed in an artificial uterus for gestation. The resulting offspring is a genetic replica of the organism that provided the somatic cell. An example of cloning done through this somatic cell nuclear transfer method is Dolly the sheep. Dolly was a sheep born in 1996 and was the first mammal cloned from an adult somatic cell, and is considered as a significant achievement. Here's a practice question. Feel free to stop the video and take your time to answer the question.